Buenos días, good morning. Good morning, everyone. Okay, 1 minute more. Please take a seat. Okay. So welcome everyone. Welcome to the ones that are in the virtual platform. So welcome. So let's wait a little bit. Okay. Okay, I think I don't have any announcement from the organization. No, okay. So everything's fine. Good morning, everyone. So it's a pleasure for me to present to Dr. María Rodríguez as our keynote speaker for today. So Maria, that I know very well, so I know her very well, is a technical lead of the group of systems biology at IBM Research in Switzerland. And she's a physicist as a training, so she did her PhD first in the high energy physics at the Institute of Astrophysics of Paris, and then a first postdoc at the Hebrew University. So then in 2006, so she realized that she transitioned into systems biology as a postdoc at the Weizmann Institute of Science, and later she moved to Columbia University in New York. So her training is impressive, so has been supported by a number of prestigious fellowships, among Marie Curie Fellowship in Cambridge, from French Ministry of Research Fellowship, Higher Education Teaching Initiation Fellowship, Lady Davis Fellowship, so other fellowships also from Israel, from Jerusalem. She has also had extended academic visits in prestigious universities, such as Caltech University, New York University, the University of Texas, and the University of Cambridge. So since 2013, she's, as I said, technical leader of the systems biology at IBM in Switzerland, but also is associated member of the Department of Biology at ATH since 2014. She's also editor of several journals, such as Immunoinformatics and Frontiers in Systems Biology. So in recent years, her research has focused on the development of computational and statistical approaches to unravel cancer molecular mechanisms using high-throughput multiomics datasets. And lately, the usage of artificial intelligence models for analyzing single-cell and drug sensitivity data for personalized medicine. More recently, she is interested in modeling the interaction between cancer and immune responses to investigate cancer immunotherapies. So with this goal, she is coordinating two European grants for prostate cancer and pediatric cancers. So today, I think she's going to talk about this new, novel, exciting work with the title Interpretable Deep Learning for Cancer Personalized Medicine. So thanks, Maria, for accepting to come here to the ECCB, and please welcome Maria. Looking forward for your talk. Thank you.
Okay, so thank you very much. It's a great pleasure to be here in one of the first face-to-face conference after a long pandemic. So it's really great to see people and discuss interacting with people face-to-face. So thank you for the organizers, Salva and Alfonso, and all the organizing team to put together this amazing conference. With that, let's disappoint. Okay. Okay, so as Anna explained, Fatima, sorry. I'm going to be presenting the work we are doing in interpretable deep learning at IBM Research in Zurich. But before going into the topic, I would like to make you an introduction because, of course, all of you and our keynote speaker yesterday, Anna Freitas, did an amazing job at introducing why we need to use artificial intelligence for personalized medicine. But here I'm going to continue in this direction. I'm going to motivate why we need interpretable artificial intelligence. Okay, so first of all, this is the three main messages that I hope I can convey in this talk, and I wish you will remember when you come back home. First of all, I'm going to give you an explanation of why we need interpretable deep learning, and then I'm going to present two different use cases. The first one focuses on personalized drug models, and the second one on T-cell-based immunotherapies. And as Fatima explained, actually, I'm a physicist by training. I like to have theoretical models. I like to understand what I'm doing. But lately, I'm starting to do deep learning. And with that understanding, I'm gonna be talking a lot about deep learning. Sorry, maybe I'm gonna take this away. I'm gonna be talking a lot about deep learning. My point of view regarding deep learning and all mechanistic models is that all models are wrong, but some are useful. So in that respect, deep learning might be useful in certain situations, but only if we do it carefully and we know what the model is doing. And that's why I'm gonna be telling you about the things I'm telling you in this talk. Okay, so I guess I don't need to make a big introduction about the amazing, outstanding achievements of deep learning models in the last 10, 20 years. This is a very summarized, for me, in my opinion, one of the main three achievements of deep learning. There are many more. This is a very subjective list. But probably we can start in 2012 with AlexNet that revolutionized vision tasks, winning the natural image recognition competition. Also outstanding was the achievement in 2016 of AlphaGo that beat the human champion at Go. If you ask me, for me, it was much more impressive what came afterwards. After AlphaGo, we had AlphaGo 0 and Alpha 0, later on developed by Google. These models were quite impressive because actually they did the same. They just beat humans and all pre-existing models at Go, chess, and shogi, but they did it in a completely independent, in a way completely independent of human knowledge. Basically, Alpha 0, they just they taught the computer to play Go and chess just only by playing against itself. And just completely independent of human knowledge, so no access to databases of games whatsoever. Just play by itself, and in a few days, the computer beat all previous models. So this was an standing achievement. And finally, I also, of course, because we are computational biologists, coming to biology, deep learning has been a bit delayed with respect to other fields because actually we have access to much smaller data sets. But nevertheless, in 2018, we have AlphaFold, and then in 2021, AlphaFold 2, which again revolutionized the way we approach the prediction of protein folding and protein structure. So of course, deep learning, after this and many other achievements that I'm not citing in this page, is clear. Nobody will debate that the deep learning is achieving outstanding predictions. But how we are doing that is by creating larger and larger models. So this curve actually shows the exponential growth of deep learning models focused on these models focused on language processing. And I chose language processing because actually this is probably one of the largest models to date. So this plot is quite impressive because if you look, we're spanning only 3 years, from January 2018 to January 2021, so only 3 years. And the model, well, I don't know if you can look at the y-axis, but they have gone from small models of only a few 100 million parameters to this model which have 1.6 trillions of parameters. So I hope, and you can see the exponential growth doesn't seem to be near to plateauing at any point. So models are getting huge. And you might argue, who cares? I mean, models are performing extremely well, and this is no discussion about that. Models can reason, these models can understand jokes, they can understand physical equations. They are doing extremely well, but 1.5 trillions of parameters. So I would argue that there are problems with big models. So what are these problems? Well, first of all, as you can imagine, they require huge amount of data. And while we might have these amounts of data in NLP tasks, certainly we don't have them so far in biology, and I'm not sure we'll have them anytime soon. But independently of the amount of data that we need, we also have other problems. For instance, there is also a lot of ongoing debate on ethical AI about the environmental footprint of these models with, well, as probably you have read in the news, some of these models take a huge amount of electricity to develop, to deploy, and eventually to maintain. So it's also a concern that sometimes we are building models that are 10 times larger, so with 10 times or even larger amount of electricity consumed for just tiny, tiny increases in performance. So it's debatable whether this is really needed. Also concerning is that, probably maybe you have heard about it, is the lottery ticket hypothesis. This was a very intriguing paper published in 2018 and has a lot of citations. So basically the observation of these researchers was, okay, you have your huge model, which is doing great, But then what you can usually find is that subnetworks, and by subnetworks I see networks that maybe just include only 10% of the parameters that have the same accuracy, and in a few cases even higher accuracy. So let me repeat that. You have a huge model and you can find a much smaller network, so you can drop 90% of the parameters and have the same or higher accuracy. And what they show in this paper is that there is an algorithm a way in which you can find these smaller subnetworks even before training. So it's not that you need to train and then you can drop parameters, it's that you can find them before training. So that makes you question whether we really need these huge models to begin with. Of course, this is a very active debate. I don't claim here that it's solved. People have claimed that there's these winning lottery networks. Sure, they are smaller, but they are more unstable. And it's true, in some cases they have been shown to be unstable, but there's other newer algorithms that have been shown to make them more robust. So what I would say, that debate whether we need really huge models is really not over. But the last thing I would like to make, and probably the one, as you can imagine, is going to motivate the rest of my talk, is that these models, these huge models, are black boxes. And again, you might argue, okay, who cares? We have a huge model that does very well, and it's a black box model. Who cares? So again, I argue that we should care, because there might be black box models might hide many problems. The first problem, of course, hidden biases. And again, here there's a very active field of research in which people are finding, especially in biology, we have all types of biases. We have gender biases in our data, we have ethnic biases, we have socioeconomic status hidden in the data. So we should pay attention when we build a deep learning model using biological data to maybe stratified cancer patients or just to give them the right type of health insurance. We have to make sure that we are giving the recommendations based on ethical criteria. So, I mean, we are not discriminating against gender or ethnicity to begin with. And there have been examples of models discriminated based on the wrong features. For similar reasons, these huge models can hide, can encode wrong hypotheses, software errors. But more important, you have this huge model, and of course, in the end, we are scientists. We want to understand why a model is working. So we want to get some insight about the biological processes that are driving the decision. And when you have a black box, you cannot get any information whatsoever. So based on that, there is the topic that I'm going to be, the models that we'll present in this talk is just the focus in the area of interpretable deep learning, which is, as you can imagine, is a very active field of research in artificial intelligence, where the idea is that we develop approaches that either they are already transparent to begin with, or if they have not, there are ways in which you can extract insight a posteriori. So as I said, this is a very area of the field. So basically the field, there are many different approaches. So what I'm going to be mostly showing you in this talk is the first type of models in which you have the models are from construction, not fully transparent, but at least you can get some insight from construction. But also towards the end of the talk, I will also give you models, the other type of models in which the models are black box, but you can extract some insight a posteriori. Okay, so with this introduction, let me now move to the applications that I would like to show you. As I did say at the beginning, there are two main different applications that I would like to present. The first one, I would like to show you a little bit about the work, tell you a little bit about the work we have done at IBM Research about predicting models for personalized drug medicine. So let's starting with the first talk, we got interested in developing models to predict drug sensitivity. So when we started thinking about this, it was around 2018, and we were interested in identifying to build models, deep learning models, that could predict the efficacy of an anti-cancer compound on cancer cell lines. This, of course, we are not the first ones thinking about this. When we started looking into it, there were already quite a lot of models available. What we found out is, after surveying the literature, is that most models use either, of course, you have to characterize the cell type. So most models will use a single type of information. Either you just pick up the mutations of a particular cell line or patient, or you pick up copy number alterations. But there were not enough effort to build models that could integrate diverse types of information. So with this idea in mind, we gave ourselves the task of using deep learning to build a multimodal approach that could integrate diverse types of information for a predictive task, which in our case was to predict the efficacy of a compound. So what are these layers of information? So of course, first of all, we wanted to integrate information about the chemical compound. And this, well, you will see in a little bit. So we can do it through, we try to survey different approaches. You can do it through fingerprints, which are just hand-defined engineering features that you can use to describe chemical compounds. But also we use a much more, well, not recent, the SMILE representations have been around for quite some time, but there has been an interest in using it right now since the development of deep learning. So basically, a SMILE representation is a string-based approach of represented chemical compounds. So basically, you have this kind of series of strings that represent a chemical compound. The second layer of information is, of course, we need information of the molecular signatures of the cell. Here again, we decided to use high-throughput data, so we define our cell as a transcriptomic profile. And we chose transcriptomic data because, again, there has been literature that shows that transcriptomic information recapitulates is one of the most informative layers of information to predict drug sensitivity. And last but not least, we also wanted to include information, the prior knowledge that we know about this cell line. And this could be information, first of all, information about the topology of how these molecular interactions molecular components interact with each other, but also about non-drug targets. And this prior knowledge is really important because as models get larger and larger, because we have thousands of potential genes features, to 20,000 or more, we need models, in biology especially, we never have cohort sizes that allow us to train from scratch a model with 20,000 features. So we need to use prior knowledge to reduce the amount of features. And what we asked the model, so we asked two different things. First of all, we wanted the model, of course, to predict drug sensitivity on particular cell lines. But also, as I mentioned before, we wanted the model to be interpretable and to give us information about the molecular features and the chemical features that were more informative for a prediction. Okay, so this is just one technical slide. If you are not into deep learning, it doesn't matter. It's not super technical. So the only thing I wanted to show you is how we do this integration of different layers of information. So basically, Pacman is built in two different pillars. Here in the pink side, is the module that encodes the molecular information and transcriptomic profiles using this prior knowledge here. And then in the second module, we have this pillar that encodes the chemical information of the compounds. And there they are concatenated, and importantly here, we use attention mechanisms to get this interpretation that I'm going to show you in a couple of slides. Okay, so let me show you some results. So of course, we did 3 different types of validation. Maybe sorry, we cannot see the distributions here, apology about it. So maybe this is one of the best ways to look at the performance of the model is to compare the predicted IC50 value versus the experimentally measured IC50 value. And these are the distributions that you should be seeing here that unfortunately you cannot see. But well, actually you can see the points here in the diagonal. So in this plot here, you have the predicted IC50 values, measured IC50 values, and if your model would be perfect, so 0 errors, all models will be laying in the diagonal. So by measuring how much you deviate from the diagonal, you can assess the accuracy of your model. And as you can see, Pacman performed very well at the task of predicting IC50 values. Also, what it was very nice to see is like the year after we published Pacman, there was an independent benchmark where the authors selected 28 deep learning models for the same task, predicting drug sensitivity. And actually, we found out that Pacman was the top performer for this particular task. So that was a very nice validation, independent of us. But let's move to the area of interpretability, because this is what I've been motivating at the very beginning. So because we use attention mechanisms that, again, if you are not a deep learning expert, doesn't matter. Attention mechanism is a module that you can integrate in your network as you are training it, and then you train it normally. And what it gives you actually just highlights which features were more important for a particular prediction. So what this allows you is, a posteriori, you can generate this type of heat maps. For instance, this is for chemical structure, where darker color means that the model Pacman paid more attention to this feature to make a prediction. And while this is qualitative, actually it allows you to make interesting comparisons. So for instance, here I'm showing you the chemical structure of masitinib and imatinib. Both of them are existing compounds that they are FDA approved for the treatment of leukemia. You can see they have similar IC50 values, with masitinib having a slightly better IC50 value. So for IC50, lower is better, so this masitinib is slightly better. And as you can see, actually, you look at the attention maps, it's quite interesting, because in the case of masitinib, you see that most of the attention is focused on this atom, sulfur. And by the way, I did not mention, but the only difference between these compounds is this atom exactly here. We have a sulfur here, we have a nitrogen here. For the rest, they're identical. And what was interesting is that Pacman, in the case of masitinib, most of the attention is focused here. All the other atoms and bonds have little attention. While in the case of imatinib, actually the attention is somehow distributed here and there with no winning feature. So it seems that Pacman is trying to find what is the winning feature, but does not find it. So again, this is qualitative, but just by looking at these plots, you can draw the hypothesis that maybe this atom is the one that is giving masitinib a slightly better IC50 value. And by doing this type of observation can allow you to elaborate further hypotheses that you can further validate. By the way, I'm not showing that here, but you can do the same analysis at the gene level. So Pacman can also generate a gene map where just is highlighting you have a different attention scores for different genes, and you can see which ones were more informative for your prediction. So if you would like to play with Pacman, we have made this, at IBM, we like to make everything freely available through a web service. It's free to use. You don't even need to create a login. And here you can either predict the sensitivity of a particular FDA-16 compound, or even you have the possibility, if you are into drug design, you are generating your own compound, you can even design your own compound, and then the model will just predict sensitivity on different cell lines. And also, of course, this is the type of the information that the server is going to give you. They, of course, give you a table much longer than this one, a table with predicted IC50 values. Also, you will get the attention maps both at the chemical level and also at the gene level. So you will get the genes that with higher attention scores. So if you are interested in that, please play with it and let us know. We are always happy to hear whether this application is useful. Okay, let me now move to the second example of how the type of models we are doing for that personalized drug design. So after we finish with Pacman, we move into a much harder task. We now wanted to demonstrate whether we could use deep learning to enhance drug design. And we developed Pacman RL, and in case you are wondering, the RL stands for reinforcement learning. So probably I don't need to make a very exhaustive, long introduction of why we need to build new pipelines, new computational approaches for drug design, but I find these numbers quite telling. So we are spending today, adjusted by inflation, 100 more, an amount of money 100 times larger than the amount of money spent in the 50s to develop new compounds, and yet compounds developed today are more likely to fail in clinical trials. And so we are spending more money and we are failing more. And this is despite the much larger amount of information about the molecular components of the cell, the wiring of the cell, and the biomedical properties of compounds. So that means that we are spending more money, we have more information, and yet we are failing more often. So obviously something is wrong in the way we are approaching drug design. And I don't claim that I have the answer, but what I think it is maybe one step forward could be just to develop approaches that can integrate in a more efficient way the information that is available. And with that thought in mind, we thought about using reinforcement learning. So in case you are not an expert on reinforcement learning, don't worry, I'm not going to be too technical on the details. Here I tell you everything that you need to know about reinforcement learning to follow this talk. So basically, reinforcement learning has two components. You need an agent and you need an environment. So basically, first of all, it's a set of iterative algorithms that you, through successive iterations, you can achieve very complex tasks. And basically, the way it works, you have an agent. An agent is something that at each iteration performs an action. And this action is performed in an environment, and the environment will give a reward. So you get a positive reward if the action takes you towards your goal, or you get a negative reward if the action takes you against your goal. And that's it. That's the main core of reinforcement learning. You iterate, and eventually you hope that will converge. So let's come back to the problem of drug design. How can we adapt this to drug design? So the first thing that we need is an agent. So what will be an agent for us? An agent could be something that we just give at each iteration a new candidate compound. So this is what we call generative models. It's a model that is trained on databases of existing bioactive compounds, and at each iteration give us something that looks like a bioactive compound. Okay? Now, we need the environment. It's also it's also called the critic. So before deciding what is the environment or the critic, you need to decide what is your goal. So our goal in our case is, and this is important, is to predict sensitivity on individual molecular profiles. And here I'm highlighting the word individual. So we wanted to decide an algorithm that pre-designed compounds for individual, either cell lines or patient. And the reason we decided to do that is because actually there has been, when we started working on that, there was a lot of models that are designing generative models to design compounds targeted to specific proteins or specific receptors or particular mutations. And all this is very good, of course, but the problem with this approach is that you need to know a priori what is the causative mutations in your cell line of choice or in your patient. And very often you might not know. There are many cancers for which we don't know what are the causative, the driver mutations. So we thought it was much better that we give the model a full transcriptomic profile and let the model decide what is more informative, instead of telling just target this particular receptor. So that was this logic. So basically, we want to design a compound targeting a particular transcriptomic profile. And the second goal that we have is also design compounds with low toxicity. So we are going to also be filtering away compounds that we predict are highly toxic. Okay, so with this in mind, let's come back to our reinforcement learning approach. So we have the agent, that means the model that generates a new candidate compounds at each iteration, and now we need an environment. So the environment for us is going to be a model that at each iteration for each candidate compound is going to tell us the IC50 value, the sensitivity of this compound, and also tell us whether it's toxic or not. So this is, again, as you can imagine, for this iteration, we use Pacman, the model that I presented before, in which basically you give a transcriptomic profile in a compound, it will give you the drug sensitivity. And also, in addition, we added a module that predicts toxicity. And based on that we have all the elements that we need for our reinforcement learning. Now we only have to iterate, and eventually we hope that we converge to something. So before showing you some results, I think it's useful to look at, to try to understand a little bit what this regenerative model is, how it's working. So the idea is that the generative model, so the agent that is generating a candidate compound at each iteration, what this model is doing is just sending you at each iteration to a random point in the space of possible compounds. This space is huge. I mean, there are estimations that we can have as many as 10 to the 16 candidate compounds. So of course, if you just pick randomly any random point, chances that it's a good compound are very low. So what the reinforcement learning setup is doing is just at each iteration is steering this random walk in this huge space toward areas in this random space that we hope had good properties. So this is another way of looking at it, steering the random walk toward good areas with good properties. Okay, so with that, let me show you some results. So we published this last year, and we did several case studies focused on different cancer types. Here I'm showing results for breast cancer. So maybe let's look first at this plot here on the left. So here I'm showing two distributions, the gray distribution and the red one. Let's look first at the gray. Well, first in the x-axis, here I have the log of IC50 values. So here, this area on the right side is the area where we have compounds with good sensitivity values. So basically, this gray area is the ones where we want to get most of our compounds. And let's look first at this distribution, the gray distribution. So this distribution shows the values, the sensitivity values of compounds generated by our agent, our generative model without reinforcement learning optimization. And as you can see, most of them fall in this area where they don't have good good sensitivity, drug sensitivity values, which is not surprising. The generative model by itself is just picking random compounds, and then you hope that they are good, but most of them are not good. And now the red distribution is the one that you get through the reinforcement learning optimization. As you can see, just by looking at the distribution, there is a significant shift towards the area of good IC50 values. So this was the first validation that this is working at a computational level. We can also look at some of the models of the compounds that were designed. So here we have this, and this plot shows compounds that were predicted. They are not existing compounds. They are predicted by our algorithm. So these were designed in different settings. Here, basically, they are targeting, these compounds are designed to target a specific breast cancer cell line, while here they are designed to target a group of breast cancer cell lines. And something you can do, of course, I'm not a chemist, but something you can do is you can look in the space of these possible compounds, you can look at the closest neighbors of existing compounds and try to see what you find. And of course, in this space, you can assess the distance in this space by using coefficients such as the Tanimoto coefficient that give you the distance between different chemical structures. So what's interesting, because when we look at the top 3 neighbors of this theoretical compound in this huge space, the top neighbor was doxorubicin, which is a drug that is standard, it is used, it's FDA approved, and is used for the treatment of breast cancer. So again, what it's telling you is that this approach is just designing compounds that are close enough to existing compounds, but they are different. So it seems that it's designing something that it has the right properties. Also, it was very nice to see that despite not directly optimizing for these properties, we also find very good behavior and other desired properties in candidate compounds, such as drug likeness. So this score tells you whether this looks like a drug, and we also got quite good behavior here. Solubility, and synthetic accessibility that tells you how easy it is to synthesize a compound. So again, we did not directly optimize for that, but we found that most of the compounds that we designed have good properties. So it was kind of so very nice to see. So with that, and this is, of course, at this point, it remains a theoretical study. We should do the experimental validation, which implies to synthesize these compounds, but at least at the theoretical level, we have able to design compounds with really good properties. Okay, and with that, I would like to close this first chapter, where I have shown you these models that we have designed for personalized drug modeling. And let me move now to the second part of my talk, so the second use case I would like to tell you about. And here, I'm going to be telling you about more recent work we have done to model cancer immunotherapies, more particularly T-cell-based therapies. But before going into the model Titan, maybe I can give you a super brief introduction about why we care about T-cells. So T-cells are, well, as you probably, all the immunologists here know, everybody knows here, T-cells are one of the most important components of the adaptive immune system. They have many functions, among them regulatory and cytotoxic functions. So this later property, cytotoxic function, is one of why we care so much in cancer immunotherapies. So T-cells, cytotoxic T-cells, have the capability of identifying infected cells or cancer cells and destroy them. And because of that, there has been a lot of interest in recent years to use them in adoptive cell transfer therapies, where the idea is that you take T-cells from a patient, you engineering ex vivo to learn them, to teach them how to recognize cancer cells, expand them, and reinfuse them in the patient. And these therapies come in different flavors. There are different ways in you can do that. So here I'm citing 2 of the most common, this chimeric antigen receptor, CAR T-cell therapies, where basically here the idea is that you engineer the cells to express a CAR receptor, which is a synthetic receptor that you have designed to target a particular cancer neoantigen. But there are also other flavors, like T-cell-based therapies, in which you use a native receptor, a real T-cell receptor that has been selected because it binds a cancer neoantigen. So in both cases, you just program these cells to express a receptor, either synthetic or natural, but that has been selected to target a cancer neoantigen. Okay, so what are the good sides of these therapies? So in some cases, in some hematological malignancies, these therapies have really achieved long-term remissions. And when I'm saying long-term, I mean longer than 10 years, which is unmatched by other more traditional therapies. Because of that, we have 2 FDA-approved CAR T-cell therapies, and there are more that are ongoing clinical trials. So I will expect that in the coming years, we'll have many more cancer T-cell-based therapies. Now, not everything is good. We also have problems with these therapies. First of all, is that many patients do not respond, and we don't know why. And also very concerning, some patients respond and they have strong side effects, among which high toxicity. So we don't know exactly the reason for high toxicity. They are not completely known. There are probably a combination of complex factors. One of the reasons, especially in T-cell therapies, is that T-cells can bind off-target very easily. So there is also the concern of cross-reactivity toxicity. So at IBM, we are investigating both therapies. We are developing models to understand both therapies, and mostly this is done in collaboration with Cyredia at ETH. But the work I'm going to be telling you here is mostly based on what we have done to model T-cell-based therapies. And to understand T-cell-based therapies, one of the first things we need to do is to build models that can predict the binding of T-cell receptors. So more concretely, what we want to do is to build a deep learning model that takes a T-cell receptor, so TCR for short, and takes an epitope and give us a binding probability. So this, again, another one technical slide, it's only going to be one, but if you want to hear how it works, so this architecture borrows many of the elements from Pacman, so especially the multimodal nature. So we have, again, two pillars, one that encodes the information of the T-cell receptors that we encode as a sequence of amino acids, and the second one that encodes the information about the epitopes that we, again, encode as a sequence of amino acids. And then here, this will be important for interpretability. We have these layers, that is context attention, that will tell us a posteriori what are the more important amino acids for each prediction. Okay, so let me show you some results. So the first thing we did was to predict, of course, we use using public data for T-cell receptor epitope pairs. We built Titan with training, and then we checked the accuracy. So here I'm showing 4 different flavors of the model. The details are not important. This basically, you are curious of what are these 4 versions are. There are different ways in which we can encode both the T-cell receptor sequence and the epitope. We can encode the full T-cell receptor sequence or only the variable part, or the epitope can be encoded as a string of amino acid or a small compound. So as you can see, this leads to small changes in accuracy, but overall, we are around the 75% accuracy as measured as the area under the rock curve. So it's a reasonable accuracy. This is what we thought. Of course, just looking at accuracy without having a baseline to compare is rather meaningless. So we designed a super naive, simplistic model, which is just uses, it's not even doing generalization, just doing classification, and this uses k-nearest neighbors. So we trained this baseline, this very simplistic baseline, and we use that baseline to compute the accuracy. And we were actually extremely disappointed when we saw that our super simplistic baseline was outperforming our deep learning models. Of course, at the beginning, this was disappointing, as you can imagine. But then thinking a bit more about it actually became clear to us what happened. So deep learning, as I mentioned at the beginning, performs extremely well, but you need data to train it. And as typically happens in biology, data is limited. So more concretely here, we use public data sets. We had around 50,000 receptors, T-cell receptors, but very few epitopes. So we kept thinking how we could increase the amount of data, because it seems that data was insufficient. And by thinking about it, we came to the realization, and if you think about it, a T-cell receptor is a protein, and an epitope is a string of amino acid, but because it is small enough, you can also use the similar encoding that you use for therapeutic compounds. Again, it's not a therapeutic compound, but you can encode it in similar ways with the SMILE representations. And if you do that, now you have transformed, at least at the computational level, you have transformed your problem into the problem of predicting protein-compound interaction. And for this latter problem, we have a much larger database. We have binding DB, we have millions of samples. So we said, okay, let's pre-train on binding DB, and let's see by pre-training, so it's another example of transfer learning, whether we can increase our accuracy. And actually, this is the good news. With this pre-training, we substantially boosted the performance of all our models. And again, here you have different variations because there are different settings in which you can pre-train. Again, the details are not important. Just the message here is that through pre-training or this transfer learning strategy, you can significantly boost performance of the model. Okay, so let's now look at interpretability. As I said before, because we use this context attention, we can look, as we did for Pacman, we can look and interrogate the model and say, okay, tell me which amino acids were more important for a prediction. So here I'm showing you segments of 3 different TCR sequences. They are selected to bind the same epitope. And you see, well, if you look at particular positions, in all positions there are variability, but the model is quite certain in all 3 cases which amino acids are important for the binding. So it gives high weights to these amino acids, which are identical in all sequences, and also high weight to these amino acids. So it seems that the model is certain that these amino acids that are concerned in 3 sequences are quite important to bind this epitope. Of course, we don't have the ground truth here, because for that we need the crystal structure, we don't have, but at least from a computational and chemical perspective, this makes sense, that we have 3 amino acids that they are not close to the border. So this, at least from a sanity check, it makes sense. Now, we can do the same exercise the other way around. We can fix the T-cell receptor and look at the epitope. And now here we found this. And as you can see here, there are many problems with this plot. So first of all, we got that, well, here I'm showing you only 3 epitopes that bind the same, they were predicted, that were, we selected to predict the binding to the same T cell receptor. So as you can see, but basically we got the same attention map for all epitopes. So you see the first problem is that we get a very homogeneous attention. So we get the same score over all epitopes. That's strange. Second, also, we got always the same positions. That's also very strange. We will expect that different epitopes bind through different amino acids. But also, most puzzling, we got, this is a padding score, a padding sequence that we just use to make all epitopes the same length, and also got attention, which this should not have. This doesn't have any biological information. It should not have attention. So again, we were scratching our head quite a lot with that. What eventually realized is that the model, so what I showed you at the beginning, we have a data set that is rather limited. We have around 50,000 T cell receptors, but we have less than 200 epitopes. So the model realizes quite soon during the training that there's absolutely not enough data to generalize, to build a model that can generalize towards new epitopes, because only 200 epitopes. This is not enough for deep learning. So the model realizes that, and what it does, actually, this is very clever, is transform that into a classification task. So it says, okay, fine, I cannot generalize to new epitopes, forget about that, so I'm gonna just classify the ones I have. And this is what it's doing. By selecting 3 random positions, you can classify 200 epitopes. So this is what the model is doing here. So I'm showing this negative example because actually I want to tell you how through this interpretable analysis, you can first identify the things that are working. So we can see that the model predicts quite well towards new T cell receptor, but also you can identify when there are problems. And here, when things are not working, the model is not learning to bind towards new epitopes, so you can also get insights with interpretability. And this is what I was showing at the beginning. So interpretability not only gives you new insights, but also signals potential problems. And this is also very, very important. Okay, so after showing this, I'm gonna now move to the last model I'm gonna show you today, where we have actually go one step further into try to interpret the prediction of any T cell receptor binding model. And I'm gonna tell you about the code. And this is an example, as I showed at the beginning, the example of what is called post hoc interpretability, in which now here we assume that we have a black box model. So this will be a black box model. So you have no idea what the model is doing. You have no access to the software, so you cannot look inside. And what we are going to develop is our approaches to extract insight a posteriori. But maybe just introduction why, because I keep telling you that we care about predicting the binding of T cell receptors, so maybe I can just give you a brief motivation of why we are so interested in this problem. So one of the challenges with T cell therapies is that these T cells are ubiquitous binders. They can bind, even a T cell receptor in a few cases has been reported to recognize more than 1 million epitopes. So of course, if you're going to engineer a T cell with a kind of with a T cell receptor, and you're going to infuse it into a patient, you better make sure that you know what this T cell is binding to. Because otherwise, what can happen, and unfortunately it has happened in clinical trials, it might happen that the T cells bind to the wrong target in healthy tissue and produce massive inflammation, and the patient might die. And this has happened in clinical trials. So it's extremely important to understand how these T cell receptors are binding. And also, if we can understand these rules, of course, we can engineer receptors of improved affinity. So of course, we are going to do this through the angle of interpretability, but just on the side, we like to mention also this effort we are doing in collaboration with other colleagues at IBM. So one of the ideas we had and we are exploring is just, you could in principle, of course, simulate this through MD simulations and try to predict the binding through MD simulations, molecular dynamics simulations. The challenge we have here is molecular dynamics are extremely computationally expensive, so there's no chance you could just do it for 1 million different epitopes. But what we are exploring is whether we can use MD simulations accelerated through AI to just have a much faster pipeline in which you just only simulate really short amount of time, and then you use deep learning to predict the missing time steps. But again, I'm not going to be telling you about this today. I'm going to be telling you about the work we did to interpret through the interpretability lens. Okay, so in that respect, we have developed the code. This is it was meant to be a pipeline, so it's customizable and user-friendly, and you can adapt it to your own needs. And the idea is that it can interpret any black box model that is designed to predict the binding of T-cell receptors. So how it works, so the idea is simple. So here you have your samples. So by us, in the code, samples will be a T-cell receptor coupled to an epitope. So for each one, we have a prediction, binding or not binding. So the first thing we do is we separate the pairs into binding pairs and non-binding pairs. And then we use a clustering algorithm to decide to find clusters of similar sequences. And the way we do this clustering step is for several reasons. First of all, there's a pragmatic reason. So this prediction is computationally intensive, so you don't want to do it for maybe the thousands of possible binding pairs that you might have. But also, it might be not very intuitive. If you end up having rules like 100,000 rules, it might be not very informative. So we want to reduce the number of rules, and what we are going to generate is one rule per cluster. Also, just a bit more to give a biological motivation. So we know that T-cell receptors might bind different epitopes, but we know that the host receptors that bind the same epitopes share similar patterns. So it makes sense to cluster those together and try to get a unique rule for this cluster of similar T-cell receptors. Now, about how we analyze each cluster, so what we wanted to do is to generate rules that are interpretable for us human users, because again, this interpretability should be human-centric. So for us, we humans, we understand well this if-then rule. So if you have this amino acid in this position, then you bind. If you don't have it, you don't bind. So this is the type of rules we want to generate. How did we do that? So here we are using anchors. Anchors is a model that is an interpretable model that is becoming quite popular in the interpretable deep learning community. Without going into the details, what anchors do is analyzing the neighborhood of each sample and generating this type of rules, if then. And then, of course, we visualize the rules per cluster. So let me show you some results. Okay, so this is, for instance, as I said, you can run this pipeline to any T-cell receptor focused model. So we have done it, of course, in our model, Titan, but also we have used a different model, PMTNet, which was published after Titan, and you can use it for any other model that you might have. So first of all, here you have the rules that the code finds for the different binding regions. This is the focus on the CDR2 region and the CDR3 regions here. So these are different regions of the T-cell receptors. Details are not important. And what I'm showing here is the frequency. This is a logo representation of the sequences in this cluster. So just tell you the frequency is how likely each amino acid is in this position. And I'm showing you here in the bottom of the rules, so just you can see that actually the rules are not just a frequencies realization. So the code does not predict the most likely amino acid in a position, but instead just find the amino acid that is important for this pair to bind together. Okay? So as I said, this is very customizable, so you can, if you play with that, you can use it, you can change the clustering approach, you can change also interpretable method. We use Anchor, but you can use any other of your choice, so it's quite user-friendly and customizable. And if you are interested also, you could in principle apply it to any other language-based model, so it's not even focused on T-cell receptor. Any other sequence model could also benefit from this type of a pipeline. Okay, so with that, I would like to start wrapping up. But before that, I wanted to give you, here, a few more bits of interpretability, and this is going to be rather short. I want to show you rather quickly other work we have done in the area of interpretable deep learning. So first of all, of course, the code I have just shown here. But also we have work on what we call mononet. These are structurally, architecturally constrained networks that we are investigating for the sake of interpretability. So basically what we are doing here is we build our models. So at the beginning they are dense models, but then if you want to interpret this particular layer in blue, what we do is just constrain the next layer to be monotonically related to each other. So that means they cannot have negative coefficient. There's always a monotonic relationship between the neurons here and the output. And why we do that is because actually, again, thinking about us, humans, the users of this approach, we understand monotonic relationships. We understand that if I make a big fire, there will be a lot of heat, and there's a small fire, there's a small amount of heat. So it doesn't matter the relationship is not linear, we can understand monotonic relationships. And actually, what was very interesting of this work is that, as we show in this work, that we could not only make the model partially interpretable, not completely, but partially, but also by doing this type of architecture, we could track what the model is learning on each different layer. And what we found is that the interpretable layer actually is acting as a bottleneck, just kind of condensating and eliminating useless information. Other work that we have done also is what we call FLAN. So FLAN stands for Feature-Wise Additive Model. So here that was our attempt at using deep learning to build something similar to the generalization of generalized linear models. So basically, FLAN, what the main idea here is that you use, basically, you encode independently each feature of your sample, and then you use a linear model. And by doing that, actually, you have a weight per feature, so you can get a posteriori, you can get, actually, this is a transparent, semi-transparent model, you can get information of which feature was most important for the prediction. And actually, we did some tests in different use cases. So this is one example we have done on a visual data set of skin lesions. And these are the explanations provided by FLAN. So basically, all models were trained to predict whether it was a skin lesion or not. And you can see the explanations provided by FLAN in this column were more focused on the lesion than in the other approaches we use. Sometimes the pixels that were deemed to be important were a little bit scattered all over the place. But FLAN really highlighted the skin lesion. OK, and with that now, I really would like to conclude. So I think the take-home message I would like you to take home from this talk is that deep learning is achieving outstanding predictions, but models are black boxes, and this is a problem. So the way I propose that we should move forward is to interpretable deep learning. So I don't think we are getting away from deep learning. Deep learning is here to stay, but let's make it transparent and more interpretable. And this is important both to extract new insights about biological mechanisms, but also about identifying algorithmic biases or things that are not working, as I showed you when I was showing Titan, so with the limitations of the data, things that we cannot predict because there's not sufficient data. So here I've showed you 2 use cases and 2 examples in each case. Here I present Pacman, that is our attempt at developing a multimodal deep learning model to predict drug sensitivity on individual profiles, cell lines. And also Pacman RL, where it's our attempt at designing approaches that enable the design of new compounds, individualized for each patient. And also in the area of cancer immunotherapies, I have discussed Titan, that is our model to predict the binding of T-cell receptors, and also the code, this is our interpretable pipeline to understand the binding rules of each prediction. And with that, I would like to thank collaborators and teams. So collaborators, I would like to thank Julio Saez, Sai Reddy, which we are developing all this, we have a close collaboration, and we are developing all these models for cancer immunotherapies. And also Hara Caponi, also at IBM Research. And also the team, I would like the whole team at IBM, special thanks to Amphi Nguyen, and also Anna Weber, who is here at the conference, and she has given already 2 talks on this. So she was a major contributor of Titan and Decode. So if you have specific questions, certainly you can track her and talk to her. And with that, thank you very much for your attention, and I'm really happy to take your questions. Thank you.
Thank you, Maria, for this really nice talk. So do we have questions? Questions for Maria? Okay.
Thanks. So you've shown a few examples where you find the attribution of the network to a specific feature. What about interaction between features? You know, we know biology is not that simple. It's complex, and there are interactions between amino acids or between nucleotides. Can you comment on that? And also, I think, for most of the examples, and also from the field, I feel that local interpretability, I mean, picking up the important features in a specific data point or example works great, and we have many successes. But global interpretability, I think Anchor that you showed doesn't have the same success, so maybe you can comment on that as well. Yeah.
So these are 2 very good questions. So going to the first one, so interaction between features, you're right. What I have shown before is as individual features, so that works well for images. But for instance, in FLAN, I did not show it here, but we also look, especially if you're going to predict the binding of T-cell receptors, probably a single amino acid is not important. You need to have interaction between amino acids. So for instance, in Flan, what you could do, and this we have tested, is just build triplets of k-mers of amino acids and train your model on these k-mers. And actually what we found out is by doing that, actually you increase the performance, but also comes at the higher, you have more triplets. So the training becomes more complicated, but then the interpretation was easier, more biologically meaningful. So you're right. In some cases, interactions are important. Now, your second question versus, yes, this is, I did not go into the taxonomy of interpretable models, but there's two other categorizations. It's just individual, like local explanations, so you only explain one sample, versus global explanations, where you try to explain the whole cohort. Of course, ideally, we would like to have global explanations. In practice, global explanations, I mean, you could do them. You could just generate an if-then tree of rules that explain the whole cohort, but the experience showed that these global explanations are so complex that eventually become uninterpretable. So yes, while desirable you would like to work towards the global, very often you are constrained to local explanations that hold in some neighborhoods. And this is probably a limitation of deep learning models, that the space of solution is so complex that it's quite difficult to find a global explanation.
Yeah, and why we did it, we are just trying to take a complex function and reduce it and limit it so we humans can understand it, but maybe it is a complex function.
Exactly. Maybe this is difficult to do this reduction.
Okay, thanks.
Thank you.
Thank you. It was an inspiring talk. I enjoyed it. Could you maybe elaborate, please? You talked about compounds you were looking for that targeted individual transcriptomics profiles. Could you elaborate a bit on what that means? Because it's hard for me to imagine what the objective you were looking for.
So actually, this is a question I got several times. So if you look at the literature, there are quite a lot of deep learning generative models that design compounds for a specific receptor that is known to be a driver in cancer X, the cancer of your choice. Or there are compounds that have been designed to bind this signaling molecule, so it's an inhibitor. So this is very good, and there are already quite a lot of models doing that. We did not want to do that because doing that means that you need to know a priori in your cancer type or in your patient, because in the end this is individualized medicine, you need to know what are the causative mutations or the causative drivers. And very often you don't know, you don't have this information. So in the end, there was a very interesting article a few years ago that demonstrated that even for known anti-cancer compounds, that technique for which technically we know the mechanism of action, actually the official or the non-putative mechanism of action is wrong. And the researchers demonstrated that because if you just block these molecules that are supposed to be the actors, the mechanism of action, the compound is still active. So it might happen that very often we have a compound that works, but we have no idea why it works. So because of this complexity, we said, okay, let's take into consideration the whole transcriptomic profile. If there are important mutations, this will be recapitulated in transcriptomic profile, and let's just let the model decide what is informative. So that was our logic in doing that. Thank you.
Hey, very nice talk. Hello? Is this on? Okay, very nice talk. So I have a specific question on your second project where you want to predict drug sensitivity. So I wonder, just following up this interpretability problem, so I wonder when you predict the drug, whether you also predict the mechanism of action of the drug, so that we know better what targets and how specific of the targets for individual cancers. For example, in your breast cancer example, how likely the drug is very specific to the breast cancer, or is generally, applicable for all cancers? Yeah.
So the thing we can do, so we're talking about the Pacman reinforcement learning, this approach.
Yes.
So here, the interpretability will come once you have designed your candidate compound that has the right properties. Of course, you can interpret through Pacman, because still Pacman is part of the approach. So you can decide what are the most important chemical patterns, and also what are the more important genes to make this drug efficient against this compound. There is a step, though, between looking at informative genes for the prediction and the mechanism of action, and we should be aware of that. So the only thing we are doing now is interpreting the model, and we hope, but this is our hope, we hope that these important genes are related to the mechanism of action, but it's something we have not proved yet. The fact that the model finds gene X important doesn't mean that this gene X is really involved in the mechanism of action in reality. So it might be that this gene X is just correlated to the gene that is mediating the function. So it's something we have to keep in mind, that in the end we're all interpreting the model, which is not the ground truth, it's just interpreting the model. Nevertheless, after saying that, something you can do is once you have your compound with the desired properties, you can look at the informative genes, and that can give you some hints of what are the involved pathways. But we don't go into really investigating the mechanism of action.
Yeah, sure. Thank you.
Thank you. Just the last question here, sorry.
Yeah, thank you. It was a wonderful talk. Yeah, so I was wondering, what you showed, the Pacman RL, couldn't you then basically use this to evade patent protection? So, you have a compound, you want to have that, you just modify it just a little bit so you evade the patent, but it otherwise works well in your application?
So if I understand your question, you say that by this approach to design new compounds, you could just design a new compound that has improved efficacy and it's just different than one that is protected by a patent.
Yeah.
I think that would be great if that happens, no?
Yeah. No, it looks like you could just do this.
Yeah, there certainly will be. We'd love to see that.
Okay.
So thank you, Maria. So thank you for
very
your
much.
nice talk.